Hi guys, welcome back to the lecture series. In previous video we have talked about pollen-pistil interaction and entry of pollen tubes in ovules. What happens next? To know this, watch till end. In terms of gamete transfer, fertilization can be categorized into zooidogamy and siphonogamy. It is zooidogamy when ciliated male gametes take part in fertilization and pollen tube formation is not required. Siphonogamy, on the other hand, is when pollen tube formation takes part in fertilization. Both types of fertilization can be observed in cycles. And siphonogamy is further categorized into monosiphonous, where a single pollen tube emerge out from a pollen grain, and polysiphonous, where more than one pollen tube emerge from a single pollen grain. This is observed among the members of family Malvaceae. After reaching ovary, the pollen tube enters ovule through one of the following routes. It is porogamy when the entry is through micropyle, chalazogamy when through chalaza, and mesogamy when it is through the integuments. Now, pollen tube may take any of these routes to enter into the ovule, but irrespective of that, the entry of pollen tube into embryo sac is always through one of the synergids at the micropylar end, and once inside it will release both the male gametes into the cytoplasm of synergid. Let us now look into the events of double fertilization. The pollen grain received by stigma is at two-cell stage containing a generative and a vegetative cell. After a successful pollen-pistil interaction, pollen tube will germinate and it will penetrate through style in order to reach ovary. In the due course of this journey, generative cell divides into two male gametes. After reaching ovary, pollen tube will enter into ovule and then into the embryo sac at the micropylar end. Pollen tube enters embryo sac through one of the synergid and releases both the male gametes into its cytoplasm. One male gamete moves toward egg cell and fuses with its nuclei to form diploid zygote. Another male nuclei will reach central cell and will fuse with the two polar nuclei to form triploid endosperm. There are two events taking place in this process. One is syngamy, where fusion of one male gamete takes place with the egg cell forming zygote, which is diploid in nature. This zygote develops into embryo. The process of syngamy is also known as generative fertilization in case of plants. Second event is triple fusion, where two polar nuclei fuses with a male gamete. This forms primary endosperm nucleus, which is triploid in nature. This further develops into primary endosperm cell and then into the endosperm. This type of fertilization or fusion is also known as vegetative fertilization in case of plants. Both the type of fertilization, because there are two fertilization processes taking place, we call it double fertilization, and this is a unique feature in case of flowering plants. After fertilization, there are a number of events taking place. It involves development of endosperm and embryo and maturation of ovule into seeds and ovary into fruits. Endosperm development is the first event after fertilization, and because it is rich in reserve food material, it is used to provide nourishment to the developing embryo. The process begins with primary endosperm nucleus undergoing several nuclear division to give rise to several free nuclei. This stage is known as free nuclear endosperm. Following this, there occurs cellularization and cell walls are laid down in order to make the endosperm cellular. This primary endosperm cell multiplies to form endosperm tissue. Endosperm may be completely consumed by the embryo before seed gets mature, or it may be incompletely consumed and is present in mature seed. As shown here in the example of coconut, the water of coconut here is an example of free nuclear endosperm And surrounding the water, white kernel of coconut is actually the cellular endosperm. You can also see presence of embryo here and rest of the part of fruit, epicarp, mesocarp and endocarp. Next is embryo development, which takes place at the micropylar end. Zygote is formed at the micropyle and it starts division to form embryo only when a small amount of endosperm is already formed and is made available to provide nourishment to the developing embryo. Early stages of embryo development is known as embryogeny and is similar in dicot and monocot plants. An example of dicot embryo development is shown here in the image, which begins with zygote. After division, it forms proembryo, then the globular shaped embryo, which converts to the heart-shaped embryo, and eventually it is then converted into a mature embryo. In a typical dicot embryo, there are two cotyledons and an embryonal axis. Part of the embryonal axis above the levels of cotyledon that ends into a plumule or stem tip is known as epicotyl, and the cylindrical portion that is below the level of cotyledons is known as hypocotyl. It ends into radicle or root tip, which is covered with a root cap. Monocot embryo will have a single cotyledon. This is embryo of a grass where cotyledons are known as scutellum. Scutellum lies lateral to the embryonal axis. A radicle and root cap is enclosed in a mass of undifferentiated cells known as coleorhiza. Epicotyl has shoot apex and few leaf primoria in a hollow foliar structure known as coleoptile. Seeds. These are fertilized ovules and final product of sexual reproduction. They are formed inside fruits and consist of seed coat, cotyledons, embryo axis. In some seeds, you'll also find presence of endosperm. Based on that, seeds can be categorized into albuminous and non or ex-albuminous seed. An albuminous seed will have a part of endosperm retained within them which is used up during germination of seed, as observed in the seeds of wheat, maize, barley, castor and ginkgo biloba. Non or ex-albuminous seed will have no residual endosperm and all of it is consumed during the development of embryo as observed in pea and groundnut. In some seeds like black pepper and beet, the remnant of nucellus is retained, which is known as perisperm. Integuments of ovules harden to become seed coat and micropyle remains as a pore in seed coat which provides oxygen and water during seed germination. With seed maturation, water content reduces and seeds become dry and metabolism slows down. Embryo enters dormancy if the conditions are adverse, or they germinate when there is favorable condition and moisture, oxygen and suitable temperature is provided to the embryo. There are several advantages of the seeds to angiosperms. Seeds have better adaptive strategies for their dispersion to the new habitat and it helps the species to colonize in other areas. Seeds are rich in food reserves. They have a hard protective covering that protects embryo. It generates new genetic combinations which can lead to variation in species. Seeds are the basis of agriculture around the globe. Dehydration and dormancy of mature seed will help in storage as in case of food and dispersal as in case of agriculture. Viability of seeds is its ability to retain the power of germination over the period of time so that even if it has entered dormancy, when suitable conditions are provided, it can bring life back and it can germinate into a whole new plant again. The period of viability may range from few weeks to several years. And as an example of this, we have two examples quoted. The seeds of lupine, which is scientifically Lupinus articus, was excavated from Arctic tundra and germinated after 10,000 years. Similarly, the seed of Phoenix dactylifera, which is a date palm, it was excavated from King Herod's palace near Dead Sea, and it remained viable for around 2,000 years. Fruits. They develop from fertilized ovary and contain seed. All other floral parts degenerate and fall off. Just when a fertilized ovule will start developing into a seed, simultaneously a fertilized ovary will mature into a fruit containing seed. Fruits can be categorized into fleshy fruit, where the pericarp or wall of fruit is soft, succulent and pulpy. Examples are guava, orange or mango fruit. Or fruits can be categorized as dry fruit, where the pericarp is hard, papery or dry, as observed in the example of groundnut, mustard or sunflower seed. Here are the example of fleshy fruits, and these are the examples of dry fruits. There are more ways of categorization of fruits. On the basis of the part of flower contributing in flesh formation, fruits can be categorized as false fruit and true fruit. A false fruit can also be called as pseudocarpic or accessory fruit, where the flesh of fruit is not just derived from ovary, but other part like thalamus is also involved in fruit formation. Apple, strawberry, cashew, all of them are false fruits. A eucarpic or true fruit will have fruit developed only from ovary post-fertilization. Most of the fruits are true. Mango is an example of true fruit. Pericarp or the wall of true fruit will consist of three parts. The outer skin of fruits is epicarp, middle fleshy or pulpy part is mesocarp, and the innermost part that surrounds seed is known as endocarp. As you can observe here, the difference between true and false fruit. We have a grape as an example of true fruit, where seeds are shown here in the middle, and we have the swollen ovary part. Seeds are fertilized ovules. Fruit here, this fleshy pulpy part of the fruit is shown, which is swollen ovary. And in a false fruit, we have got the edible part. The edible part of apple is actually made up of other floral parts which fuse and swell. Pericarp is here at the center, which surrounds the core containing ovary and the seeds. Another way of classifying fruits is to divide them into simple, aggregate or composite fruit. A simple fruit will develop from monocarpillary ovary or multicarpillary syncarpous ovary, where the pistils are joined. Simple fruit may be dehiscent, that can liberate seed at maturity in peas or beans. Indehiscent, where fruits do not liberate seed at maturity and they are released only after disintegration of the fruit wall, as in case of cashew nut or cereals. An aggregate fruit develops from multicarpillary apocarpous ovary, where the pistils are free. And raspberry, strawberry, blackberry, all of them are example of aggregate fruit. A composite fruit, also known as multiple fruit, will develop from the complete inflorescence, as in case of jackfruit, pineapple, mulberry or fig. Another term is parthenocarpy, where fruit formation takes place without fertilization and it can be induced by the application of growth hormones. Here the fruits will be seedless and they are known as parthenocarpic fruits. Banana is a classic example of parthenocarpy. Here are the examples of simple fruit, aggregate fruit, and multiple fruit. There are two more terms to discuss: apomixis and polyembryony. Apomixis is a form of asexual reproduction that mimics sexual reproduction, and seeds are produced without fertilization. This is observed in some members of Asteraceae and grasses. There are many ways to achieve this. A diploid egg cell can develop into embryo without getting fertilized, or the nucellar cells surrounding embryo sac can start dividing and protrude into the embryo sac and develop into embryos. Usually there is a single embryo in each seed, but in some cases a seed may have more than one embryo, and this condition is known as polyembryony. There are two types: simple polyembryony, where due to fertilization of more than one eggs result into occurrence of more than one embryo in a single seed. Examples are Cycas and Pinus. On the other hand, adventive polyembryony is due to formation of sporophytic budding that result into formation of extra embryos, as seen in citrus fruits like oranges and lemons. That is all in this chapter. Next chapter is on human reproduction. Stay tuned and keep watching. Thank you.